Alright, what's going on guys? ErickMed here, and in this video I'm going to be breaking down a section bank passage from the AAMC website. Now if you go to the AAMC, they have a bunch of stuff, to prepare for the MCAT. They have, you got to buy it, and you get like question packs, practice tests, flashcards, all that stuff, and you get a section bank. And the section bank is known to be of a higher difficulty than the other practice materials. And in this one, this passage, it's going to be, this is a section bank passage. This is passage 6 taken directly from the section bank. Now you do have to pay for these, so I'm not sure how long that this video is going to be up for, but this is the last one I'm doing. I have 2 other videos on, breaking down section bank passages. So go ahead, and before I begin to break down everything, go ahead, go through this passage. I'm going to scroll down for you guys. So go ahead, go through this passage, pause it when you need to, and go through these questions. This is question number 1, question number 2, number 3, 4, 5, 6. Yeah, that's it. Okay. So go ahead, go through these questions, write down your answers, and hopefully, you get everything right. So let's begin. Passage 6. Second messenger CAMP activates protein kinase A, which can then activate nuclear transcription factor CAMP response element binding protein, CREB. Alright, just by looking at this first sentence, I already know this is a signal transduction pathway. Okay, and this is actually really good because that's something that's very high yield in the MCAT. Okay, signal transduction pathways is very high yield. So if you don't know what signal transduction pathway is, I highly recommend you go ahead and review all that stuff. It's like just as high yield as amino acids. It's very, very important stuff. And basically, a signal transduction pathway, quick lesson, a ligand, which is a primary messenger, binds to a receptor. This triggers a cascade of events. A bunch of phosphorylations take place. Bunch of series goes on, a lot of reactions, and then eventually it goes to the nucleus, and a transcription is altered. Transcription can be activated or it can be blocked or whatever, but it creates a cellular response. Okay, so go ahead, if you didn't know this signal transduction pathway, if you don't know it, then go ahead and review. So let's continue. CREB exists as 2 isoforms in cells. One being CREB 327 residues. Okay. It tells me it exists as 2 isoforms, one being CREB 327. It doesn't really tell me the other isoform. That's interesting. But let's continue. CREB 327 is phosphorylated by glycogen synthase kinase at serine 115. pKa, protein kinase A, phosphorylates CREB 327 at serine 119. All right. Also, it's very important to know in the MCAT, you're going to encounter a lot of these, it phosphorylates at this position, all this stuff. Phosphorylations, it's very common. Basically, phosphorylations add a phosphate group, and it's a protein can be quote unquote activated. Okay. So variants of wild-type CREB 327 that cannot be phosphorylated at serine 115 or 119 were created. CREB 327 115 and CREB 327 119 respectively. So variants of this wild-type of this protein, 115 and 119, they can't be phosphorylated at these positions. All right, so nothing really complicated here. So experiment 1. CREB 327 wild type and CREB 327 119, remember 119 is the one that can't be phosphorylated at position 119, were incubated with various combinations of pKa and GSK3 in the presence of radio-labeled ATP. SDS page gel electrophoresis was performed under reducing conditions, and radio-labeled protein bands were detected. And these radio-labeled protein bands, let's see, they represent phosphorylation. So in experiment 1, what's going on here is that we have CREB 327 wild type and CREB 327 119. 119 can be phosphorylated at position 119. And this experiment involved getting Crab 327 wild type and Crab 327 non-19, throwing pKa and GSK3, and then with just GSK, with just pKa, and then with both, and they saw, okay, what gets phosphorylated? Does it get phosphorylated? And I'm not going to go into like the specific details of this figure because I don't want to waste time. Whenever you're going through these passages, only dissect figures and tables if the question asks to it, because that's how you save a lot of time. So we're going to keep going here. Experiment 2. Crab 327 wild type was incubated with radio-labeled ATP in either pKa, GSK3, or sequentially with pKa and then GSK3. So kind of like the first one. Isoelectric focusing and audio radiography were used to detect phosphorylation. Okay, so again, it's kind of similar here. They got just Crab wild type, and they threw pKa, GSK3, these kinase enzymes that phosphorylate the Crab, and then they did pKa and then GSK3. But instead of SDS page, they just did isoelectric focusing. And isoelectric focusing is, I would say, yeah, it's pretty high yield. You got to know isoelectric focusing. It separates proteins based on the isoelectric point. Okay, so that's what they did here. I'm not going to go through this, save some time. Experiment 3. A specific assay uses the percent conversion of chloramphenical, I butchered that, CHL, to its acetylated form CHLA as an indicator of the transcriptional activity of Krebs. Okay, so let's see. Okay, a little complicated here, but not really. CHL, when this gets acetylated, it's an indicator of the transcriptional activity of Krebs. So when we're acetylated, we have transcriptional activity of Krebs. So it's a good indicator. So I'm just going to highlight this. Here. Why am I highlighting it? Well, because as this passage, I can see there's a lot of details here. We have this Krebs, and then we have this phosphorylation at 115 for just GSK3, and then we have pKa at just 119, and then we have this, and then this, and then it could get a lot of details. A lot of details are coming in here. So I would just highlighting this so I can kind of make a mental note here saying that when we are acetylated, when CHL is acetylated, this is good. This means we have transcriptional activity of Krebs. And a lot of times questions will ask, like, okay, if this is acetylated and if this gets knocked out, if we use this instead and this goes up, and if this goes up and this decreases, and if this decreases, what happens to this? And a lot of questions will be doing that. So I'm just made a mental note here and just highlighted this just in case. All right, plasmids encoding Krebs 327 wild type and its variants were transfected into cells, and their transcription activities were measured. So the wild type and the variants, what were the variants? The variants were the 119 and the 115 that can be phosphorylated at those positions. So they got those, transfected them into cells, and measured their transcription activity. And this is the table that measures their percent conversion of CHL to CHLA of the acetylation. And that corresponds as an indicator of transcriptional activity. So not bad stuff here. This is actually a really good passage. It covers a lot of high yield stuff. Like SDS page is definitely high yield. Isoelectric focusing, phosphorylations, all this secondary messenger stuff. So that's pretty good. This is a good passage. I like this. So let's go to the question. The method used to visualize Krebs 327 wild type in experiment 2 must include which step? Again, when you're reading the questions, read the question. Literally just read the question, okay? The method used to visualize Krebs 327 wild type in experiment 2 must include which step? Let's go to experiment 2 and only experiment 2. So let's go to experiment 2. What do we have here? This method was isoelectric focusing and audio radiography. Isoelectric focusing. This is okay. So this is like a kind of pseudo discrete question. You got to know your background content here. You got to know your isoelectric focusing. You got to know your protein separation techniques. You got to know your isoelectric focusing, your anion, cation exchange, SDS page, electrophoresis, all that stuff. You got to know it. That's all high yield stuff. So this question's like, okay, what's isoelectric focusing? Amplify the cells by PCR before running on the gel. Okay, that's in gel electrophoresis. We do that. And I don't know, you would never do this in isoelectric focusing. That's completely different. Isoelectric focusing separates on isoelectric point. An isoelectric point is when the protein is neutral. And the protein becomes neutral by traveling in a pH gradient. So like if a protein was negatively charged at neutral pH, so at pH 7, if a protein's negatively charged, it will travel to the positively charged. It'll go to a pH where it can become neutral and stay there and not move in the pH gradient. That's its isoelectric point. So I knew that from my background knowledge. Establish a stable pH gradient in the gel before adding samples. That makes a lot of sense. Denature the samples so the proteins unfold into a linear chain. That's SDS. That's sodium diisosulfate that does that. So this is a no. This is a no. Treat the samples with a chemical that will add an overall negative charge. That's literally SDS. That does that. So this is a pseudo-discrete question. Basically, what the hell is isoelectric focusing? And it's B. So this should be correct. Okay. Come on, give me a harder question. What was that? Which conclusion about the phosphorylation of Krebs 327 wild type is supported by the data in figures 1 and 2? Okay, this is better. Now we got to dissect these figures. What can I see? What I don't even have to look at the answer choices. Let's make a conclusion. Figure 1. I should know the answer before I even check the answer choices. So let's see here. Here we have Krebs 327 wild type, and here we have Krebs 327 119. Let's go lane by lane. Okay, so here, GSK3 plus pKa, the two kinase enzymes that phosphorylate. Do they phosphorylate Krebs? Well, there's no Krebs added, so they shouldn't be phosphorylating anything. Boom, second lane. Here we have, okay, in these next lanes, we just have the wild type. So with the wild type only, and GSK3, there's no phosphorylation. Wild type with pKa, we have phosphorylation. Okay, interesting. Wild type with pKa, with GSK3, we have even a thicker band here. Okay. Let's check Krebs 327 119. With GSK3, nothing. With pKa, nothing. With both, nothing. So this wild type gets phosphorylated with pKa only, not GSK3. However, when pKa is with GSK3, when they're together, boom, we get this. So I'll repeat that. GSK3, no phosphorylation. GSK3 with pKa, we get phosphorylation. pKa alone, we still get phosphorylation, but not as big as with GSK and pKa. Okay. So let's look at figure 2. This is just isoelectric focusing. They're separated. pKa, again, we have phosphorylation here. pKa, then GSK3, boom, we have a thicker band here. All right, it's pretty simple. So I already made a conclusion in my head. I should find an answer choice that goes along with it. Which conclusion about the phosphorylation of Krebs 327 wild type is supported by the data in figures 1 and 2? Again, just the Krebs 327 wild type. GSK3 also phosphorylates Krebs 327 wild type at residue 119. This is not, no. First of all, they told us that GSK phosphorates at 115. So this is not supported at all. This is no. GSK3 phosphorylates Krebs 327 wild type at a faster rate than pKa. Okay. No. They talked about something, percent conversion in table 1, but they're talking about figures 1 and 2. Boom and boom. 1 and 2. This is kind of about like table 1, which is down here. It's not these two. So you go away. Phosphorylation of Krebs 327 wild type by pKa occurs at more than one residue. Do I Do I have information to support that? No. Phosphorylation of Creb 327 wild type by pKa occurs at more than one residue. It could be true. I mean, there are two residues. There's 115 and 119. Maybe. But that's not what these figures are talking to me about. These figures want to know about phosphorylation of Creb wild type. If GSK or pKa is better, which is good? GSK, pKa. What about both? What happens when we put both in there? This is not much. There's not much evidence to support this. So this is not a good answer. But I won't exit out. Let's look at D. Phosphorylation of Creb 327 wild type by GSK3 can only occur after phosphorylation with pKa. That's a good answer. Why is this a good answer? Because it's kind of like a conclusion I was making before I even looked at these answer choices. Okay. No GSK, nothing. pKa, something. GSK with pKa, big or something. Phosphorylation of wild type. Here's a wild type. So we're looking at these over here. This is the 119. So we're just looking at these. Phosphorylation of Creb 327 wild type by GSK3 can only occur after phosphorylation with pKa. Yes, this is very good answer. This is no phosphorylation. And then when we have pKa, we do get more phosphorylation. So this is very supported. So I'm going to go with D. Boom. Again, as I'm going through these answer choices and questions, you guys can see how simple it is. This is supposed to be hard. You're a section bank. Be hard, You not be hard. This is easy. But I'm very confident in my decision making, okay? That's the thing. You have to be confident. Don't convince yourself. Don't stay on an answer. Because look, I could have stayed on this answer because it's kind of supported. It does get phosphorylated at 119 and 115. GSK phosphorylates at 115. pKa phosphorylates at 119. But this is a better answer. This is barely good answer, but this is very supported. This is not really supported. This is very supported. I could have stayed here and read this answer choice and convinced myself that this is a good answer choice, but no, this is a good answer choice. I'm confident this is good. You got to be confident. All right, let's go. When designing variant Creb 327-119, which of the following residues was most likely chosen as a substitute for residue 119 in Creb 327 wild type? Okay. Remember, Creb 327-119 cannot be phosphorylated here. This variant is created, this Creb 327-119 variant is created because it can't be phosphorylated at 119. So they substituted something so it can be phosphorylated. So which of these amino acids, when we substitute it, prevents it from being phosphorylated, prevents Creb from being phosphorylated? This is background. This is pseudo-discrete question. You got to know which amino acids can get phosphorylated and which amino acids can't get phosphorylated. Tyrosine has a hydroxyl group. All amino acids that have hydroxyl groups, that have OH group, can be phosphorylated. Tyrosine has an OH, so you're done. Glutamate, it doesn't have an OH, so you're done. Alanine doesn't have an OH. Wait, hold on. Backtrack what I said. Glutamate does not have an OH, so it could be substituted because it prevents phosphorylation. Alanine does not have an OH, so it can prevent phosphorylation. Threonine does have an OH, so it can be phosphorylated, and we're looking for something that's substituted so it can't be phosphorylated. So this is gone. Okay. They left me glutamate and alanine. I'm looking for an amino acid that can not be phosphorylated. So which of these two amino acids is more likely to not be phosphorylated? Interesting. Well, I remember the side chains of glutamate and alanine. To be honest, alanine is boring. Alanine does not have like the side chain like glutamate does. Alanine is just branched with just like methyl groups, whatever. And glutamate is just not. Here, look. I'm pretty sure it's alanine because I'd rather substitute alanine in than glutamate. Because alanine is more like, here, look, watch. So I'm going to search up alanine just to double check with you guys. Oh, not alanine aminotransferase. Whoops. Yeah, look at this alanine. Boom, it's just a CH3 here. And then if I look at glutamate, yeah, we have oxygens here. Yeah, alanine is way more likely to not be phosphorylated just by looking at it. Look at that. You know? So it should be alanine. Yeah. So know your amino acids, guys. Know your amino acids. It's very high yield stuff. So that was a pseudo discrete. So very easy stuff. Come on. Which statement explains how two forms of Krebs can be generated in cells? Okay, they told me something about the forms of cells. Let's see, where is it? Okay, here. Krebs exists as two isoforms in cells, one being Krebs 327. Isoforms. Isoforms. Which statement explains how two forms of Krebs can be generated in cells? Okay, pre-mRNA is transcribed from one Krebs allele versus the other. Nah, that's not how isoforms are made. Not by the allele. Isoform is talking about the protein. Krebs is a protein. Krebs is a binding protein. They tell me that. So how can two different forms of a protein? How can there be two different forms of a protein? Not by this. This is not it. So go away. Krebs mRNA transcripts with different combinations of exons are generated. That makes sense. You could have different exons. Okay, this requires some background knowledge. Gene expression. Okay, the central dogma is DNA becomes RNA. RNA becomes mRNA, gets spliced out, introns get spliced out, whatever, and then mRNA gets translated into a protein. We can make different forms of protein with different exons. So this makes sense. Different post-translational modifications of Krebs occur in the endoplasmic reticulum. Okay. Two different forms of proteins can be made from post-translational modifications. However, this is a transcription factor. This could be it, but I feel like this is a little better answer. But we'll stick with B and C here. We won't eliminate C yet. Pre-mRNA of various lengths are transcribed based on different termination sequences in the Krebs gene. Pre-mRNA of various lengths are transcribed based on different termination sequences in the Krebs gene. Krebs is more of a transcription factor. It activates transcription of the gene. Why? No, see, this is not it. That's not how the central dogma works. RNA polymerase, okay, that transcribes the gene. And I don't think, and I've never heard of RNA polymerase transcribing one section and then says, okay, I'm gonna go for a break and then transcribe this section. No, RNA polymerase transcribes. Boom, it transcribes. It doesn't like just transcribe some of a gene and then goes wherever and then transcribes the other part of the gene. No, RNA polymerase transcribes usually in one motion. It doesn't like go out and terminate in different parts. And I don't know, various lengths. That's not a good answer. I know what I know, and I don't know what I don't know. Okay? From what I know from undergrad and the Kaplan books, okay, this is actually a really good example here. From what I know from the Kaplan books and undergrad, they never told me that RNA polymerase can jump and transcribe, wherever it wants. No, RNA polymerase finds that promoter and then it transcribes in one show. So this is wrong. So I'm going to X that out. So now I'm left with 2. This is such a good answer because this is proven. I remember reading about this. Different proteins can be formed if we get rid of an intron, if we have alternative splicing. That's what it is. If we have alternative splicing of exons and introns, because some exons can get spliced out if you are spliced at different intron locations. If you skip an intron and some exons, it could be different combinations. That's where alternative splicing is. So this is making sense here. And also residues here. They told me that it's 327 residues. Different post-translational modification. Yeah, this is such a better answer. Forms of Krebs. Isoforms. Iso means same. Yeah, so I'm going to go with B. It's a better answer. And also, post-translational modifications in the endoplasmic reticulum. That's not a good answer. This is the best way that you have isoforms of proteins, not through endoplasmic reticulum modification. Isoforms is made from alternative splicing, so I'm going to go with B. That's my answer. Boom, let's go. Which statement about Krebs 327 phosphorylation is best supported by table presented at table 1? Okay, let's go. Table 1, only table 1. Let's look at what happens here. So remember, as I highlighted here, transcriptional activity corresponds with the conversion of CHL to CHLA. When we get acetylated, we got that transcriptional activity. What gets the most acetylated? What gets the most converted? Krebs 327 wild type. This one's second, this one's third, and this is the control. Okay, so let's see. Phosphorylation by GSK3 has no effect on Krebs 327 wild type activation levels. No, this is wrong. Why is this wrong? Because look, remember, 119, these forms mean that we can't be phosphorylated at this position. 119 means we can't be phosphorylated at 119. 115 means we can't be phosphorylated at 115. So what was GSK? Is that 115 or 119? Let's see. Hold on. GSK was 115 and pKa was 119. So when we're not phosphorylated at 119, we suck. We're bad. We only have 1.7% conversion. When we are phosphorylated at 119, but we're not phosphorylated at 115, we do better. We got 8.5. And when we're phosphorylated at both 115 and 119, which is aka the wild type, boom, 27. That's good. And control, when we're not phosphorylated at 119 and we're not phosphorylated at 115, we suck. So more phosphorylations, the better we are. The more activation. Makes sense. So they're telling me that GSK3, which phosphorylates at 115, has no effect. It does have an effect because this is without GSK and this is with GSK. Look at that. We go from 8.5 to 27 when we have phosphorylation of GSK. So this is wrong. pKa has a higher specificity. What? That's specificity where? What? Like, we're not talking about specificity here. This is not supported by this data table at all. Go away. Phosphorylation by pKa can partially activate Krebs 327. Okay, well, pKa was 119. This is no pKa. This is no phosphorylation by pKa. And this is with phosphorylation by pKa. Remember, pKa phosphorylates at 119. GSK phosphorylates at 115. So this is no phosphorylation of pKa. This is with phosphorylation of pKa. When we have phosphorylation of pKa, which is 8.5, then we're more activated. Why are we more activated? Look, 1.7, no phosphorylation of pKa, we're at 1.7. With phosphorylation of pKa, we're at 8.5. We do get activated more. We go from 1.7 to 8.5. That's good. This is a good answer. Let's check D. The KCAT for the phosphorylation of Krebs 327 by GSK3 is higher than the KCAT. Okay, KCAT, those are equations. Okay, I like when I see KCAT and KM and VMAX, I like to see numbers. Okay. They're only telling me about KCAT here. But KCAT is the turnover number. And I don't see a VMAX here given to me. I don't see a KM given to me. I need support to talk about KCAT. Okay, so you go away. So this makes sense. Eliminate everything else. So I'm going to go with table 1. I'm going to go with C. Boom. Awesome. Let's go. Next one. Oh my God. We're late. Okay, let's go. Keep going. Under certain conditions, PKA and GSK3 have been shown to autophosphorate. Okay, so this means they don't phosphorate our Krebs, and they just phosphorate themselves. The control group used in experiment 2, lane 1. What's experiment 2? Okay, experiment 2, lane 1. Was designed to account for this possibility. Given this, the control group most likely contained. Okay. All right. PKA and GSK3 are kinase enzymes. They go ahead and phosphorate stuff. They phosphorate Krebs specifically. So I want to look for something where Krebs is not phosphorylated and PKA and GSK3 are phosphorylated only. A Krebs 327 wild type with ATP but no PKA or GSK3. Krebs 327 wild type with ATP. No. Krebs gets phosphorylated by PKA and GSK3. If PKA and GSK3 autophosphorate, that means they phosphorate themselves, not Krebs. You go away. PKA and GSK3 with ATP but no Krebs 327. That makes sense. They phosphorylated themselves. They didn't phosphorylate Krebs. They phosphorylated themselves. So PKA and GSK3 should be phosphorylated. They should have ATP. They should have phosphate groups. Makes sense. PKA and GSK3 with no ATP or Krebs. No ATP, that means they didn't autophosphorate. What does that help? That doesn't help us at all. They're telling us that they autophosphorate. So you go away. Krebs with no ATP, PKA, or GSK3. No ATP, PKA, or GSK3. That doesn't really tell us much. They autophosphorate. We're talking about PKA and GSK3 autophosphorating themselves. So we want to see something where PKA and GSK3 have phosphates on them. If we don't have ATP or GSK3 or PKA, what are we measuring here? We're measuring nothing. So you go away. This is a better answer. Boom. Let's go. Oh, and that was it. All right. Well, thanks for watching, guys. I appreciate it a lot. As you can see here, like I've broken down this passage and then I go to these questions and like I talked about these questions like they're easy, you know? It's easy to get lost in the information here that they're presenting you and the phosphorylations and, you know, GSK, PAK, all this stuff here. But if you break it down, you know what you know and you don't know. Again, the MCAT is a big dog, big bark, but no bite. Okay, this is a lot of barking here. It's a big bark. You know, isoelectric focusing, audio radiography. Like, why you gotta throw this in here? You know, percent conversion of acetyl, chloromethylphenol, whatever that is. Like, they're trying to bark at you. But as you can see here of these questions, literally, it's not that bad, these questions. Like, this is literally a pseudo-discrete. No isoelectric focusing, that's it. Look at the figure. Thin band, thick band. No thin band, big thick band. That's easy. Know your amino acids. You should know your amino acids like the back of your hand. Use Anki for that. I'll make a video on about how I make Anki cards and stuff like that. That video is coming out soon. Don't worry. Boom. Know how to make isoforms. Isoforms are made by alternative splicing. Just literally look at this. Look at table 1. This is probably the most trickiest question. You guys probably got this one wrong. But again, you got to know what 115 and 119 are. And they tell you. If you forget, if you're unsure, you go back. And this is an easy one. You just got to know how experiments work here. Phosphorate themselves. And yeah, that's it. All right. Thanks for watching, guys. Like, share, subscribe. I know it's a pretty long one. I'm going to make a video about Anki cards soon. Let me know what you guys think. If you want another video like this, like, share, subscribe, do all that fun stuff. And I'll see you guys in the next video. All right, peace.